How does cancer's underlying biology work? Cancer is a complex disease that starts when normal cells undergo changes that disrupt their growth control. This process can be thought of as a series of steps where a single normal cell transforms into a malignant tumor. It usually involves multiple genetic mutations and the selection of more aggressive cell clones over time. At the heart of cancer biology are two main types of genes: oncogenes and tumor suppressor genes. Oncogenes are mutated versions of normal genes that promote cell division and survival. Examples of these include MYC, RAS, and HER2. On the other hand, tumor suppressor genes like TP53, RB1, and BRCA1/2 normally help control cell growth by repairing DNA or triggering cell death when necessary. When these genes are mutated, cells can grow uncontrollably. Cancer cells often show genomic instability, which means they accumulate mutations at a higher rate. This instability arises from defects in DNA repair mechanisms. For instance, when the BRCA1 or BRCA2 genes are not functioning properly, DNA damage cannot be repaired effectively, leading to an increased risk of cancer. This allows cancer cells to evolve quickly, gaining traits that help them grow and spread. In reproductive cancers, such as testicular and cervical cancers, these principles still apply. Cervical cancer, for example, is often linked to persistent infections with high-risk human papillomavirus, which can disrupt tumor suppressor proteins. This disruption leads to abnormal cell growth. Testicular cancer involves genetic changes that affect the growth and differentiation of germ cells. The development of cancer can be broken down into stages. First is tumor initiation, where a genetic change in a single cell leads to abnormal growth. Next is clonal expansion, where this mutated cell divides, creating a group of identical cells. Then comes tumor progression, where additional mutations occur, allowing for the selection of more aggressive cells. Finally, invasion and metastasis happen when cancer cells spread to other parts of the body through blood or lymphatic systems. At the cellular level, cancer cells bypass the normal controls of the cell cycle. Normally, proteins like P53 act as checkpoints to repair DNA or trigger cell death if the damage is too severe. In cancer, these checkpoints fail, allowing damaged cells to keep dividing. Cancer cells often activate telomerase, an enzyme that extends the ends of chromosomes, enabling them to divide indefinitely without aging. In summary, cancer's underlying biology involves the breakdown of normal regulatory systems in cells due to mutations in oncogenes and tumor suppressor genes, genomic instability, and the ability to evade cell death. These mechanisms lead to the uncontrolled growth and spread of cancer cells, including those found in reproductive cancers like testicular and cervical cancer. Understanding these processes is essential for developing targeted therapies and improving early detection strategies. Always consult healthcare professionals for personalized medical advice.